Hi, my name is David Jarvis. I'm excited to talk to you today about the research that I'm doing at Brigham Young University to develop genomic and genetic resources for the agronomic improvement of quinoa. Everything that I'll present today represents work that is done in collaboration with Doctors Jeff Mon and Rick Jelen, also here at Brigham Young University, as well as Dr. Mark Tester and his group at King Abdullah University of Science and Technology in Saudi Arabia. So of course, we know the world population is growing. And unfortunately, out of 30,000 known edible plants, we're currently producing about 60% of the energy for the world food population from just 5 cereal crops. So we need to produce not only more food to feed a growing world population, but we also need to produce more food from a more diverse set of crops. And this is going to require new, hardy, highly nutritious crops that can provide the necessary calories and nutrition for a growing world population, but also grow in lands that are becoming degraded due to years and years of agricultural production. And so quinoa has emerged as one possible crop that can be used to help improve global food security. And that is because, first of all, quinoa can grow on these marginal lands. It can grow in places where many of the major crops today can't be grown. This is a picture of quinoa growing near the salt flats in Bolivia. And it's growing in very sandy and very salty soils that wouldn't support growth of most crops, but is still able to produce a crop here. In addition to being hardy and able to grow on marginal lands, quinoa, of course, is also highly nutritious. So in comparison to other major cereals, quinoa has, for example, a higher or comparable protein content. And within that protein, quinoa also has an optimal balance of the essential amino acids that we need to consume in our diet. For example, lysine, which is deficient or below the recommended standards in many of the other cereal crops, is higher in quinoa. So we're interested in being able to grow quinoa for a growing world population. Unfortunately, global quinoa production is low and is mostly centered in countries in the Andean region of South America, including Peru, Bolivia, and Ecuador. Global production is around 100 to 200,000 tons of quinoa per year. Just to put that into perspective, here is what global maize or corn production looks like. So it's grown throughout the world, and maize production is more than 1 billion tons per year. So we would like to be able to expand quinoa production into regions outside of its native Andean region. Unfortunately, quinoa is well adapted to grow in the Andean region, but when we try to grow it in other places, it is not as well adapted. So this, for example, shows some of the problems that we face in growing quinoa in different locations throughout the world. Here is a quinoa seed head that received late season rainfall. And the seed, which had not yet been harvested on the seed head, began to germinate. So you can see the quinoa roots beginning to emerge from these seeds that are still on the plant. So this would be a loss of these seeds. Seeds also shatter and fall from the seed head and fall on the ground. So you can see all these white specks on this photo showing quinoa seeds that have fallen to the ground and can't be harvested. And quinoa is also susceptible to a number of diseases and pests that it may not naturally be tolerant to. This shows quinoa that has been attacked by a fungal pathogen and resulting in loss of the seed of this plant. It's also susceptible to new insect pests. This shows pictures of plants growing in the Pacific Northwest of the United States. Here's a picture of a leaf being eaten. There are also stem borers that can bore into the stems of quinoa plants, causing them to wilt and eventually die. So as we grow quinoa in new environments, we need to be able to increase its resistance to some of these pests and diseases and address some of these other agronomical issues. And so to do that, our work focuses on developing genomic resources and genetic resources that we can use to try to improve quinoa production in new environments. And so I just want to quickly give you a few examples of each of these from the research that we're doing, beginning with some of the genomic resources. So the first one major genomic resource that we produced was a genome sequence of quinoa. So we sequenced the genome of the coastal Chilean accession known as QQ74. And this is a picture of a representative QQ74 plant. The sequencing was done with a high quality sequencing technology called Pacific Biosciences, or PacBio sequencing. So all genome sequencing produces sequences that are smaller than the actual chromosomes. And so these sequences have to be put together and assembled. And so we helped assemble them using scaffolding with different types of maps, including optical and high-C maps, to try to make these assembled pieces as big as possible. We ended up assembling about 1.39 billion base pairs, or gigabase pairs. This represents most of the estimated 1.45 billion base pair genome. So we were able to sequence most of the genome and assemble it, and identify about 45,000 genes in the quinoa genome. Despite our best efforts to assemble the pieces into as many large pieces as possible, we still ended up with thousands of small pieces of DNA. So quinoa is a tetraploid with 18 chromosomes. So it gets 2 sets of chromosomes from mom and dad. Ideally, then we should be able to put the genome together into just 18 really large contiguous sequences representing the 18 chromosomes. So although this genome was very high quality, we were clearly a ways off from assembling those 18 big pieces. We had more than 3,000. And so to try to further put those together, we anchored hundreds of those scaffolds to what is called the genetic linkage map to understand how they go together. And this produced 18 large sequences, which we refer to as chromosomes, although they may not necessarily represent the entire chromosomes. And out of the entire genome sequence that we produced, we were able to anchor about 85% of it to these 18 chromosomes. And so this was an initial high quality genome sequence that we produced, but we knew that there were problems with it. This shows a comparison of the 18 chromosomes representing the 2 different subgenomes shown in blue and green because it's a tetraploid. And the lines connect similar genes on the 2 different sets of chromosomes. And so because these chromosome sets are related, we should see a high degree of collinearity between these genes on the different chromosomes. And that's what these lines represent. And for some chromosomes, it was clear that they had been assembled quite well and put together well. But it was also obvious that we had some mistakes. Some chromosomes were too big. For example, this chromosome, which we called chromosome B1, appeared to represent a fusion of 2 different chromosomes. Other chromosomes were smaller than we thought they should be, as shown here. And clearly these chromosomes look like they belonged together with other chromosomes. And so we knew that some of the sizes of these chromosomes was not reflective of their true biology. And this was further evidence when we compare to another related species. So this is showing again the 2 sets of chromosomes in quinoa, the A set and the B set, and showing how the genes line up related to another species shown in the middle. This species is sugar beet, which has a high quality genome produced for it with 9 chromosomes. And so if these genomes are related and are assembled correctly, we should see a high degree of collinearity of these lines connecting both chromosomes. And for some chromosomes, that's the case. We can clearly see, for example, this chromosome A4 in quinoa matches up very well with chromosome 2 in sugar beet. But other chromosomes were obviously had issues. Here's that really big chromosome again in quinoa, which again appears to represent 2 different chromosomes that were incorrectly put together. And here's some of those small chromosomes in quinoa, which again look like they should actually be combined with other sequences to represent a larger chromosome. So although we were happy with this first effort at producing a quinoa genome sequence, we wanted to try to improve it. And so we did what we call a version 2 genome assembly, where we took the existing PacBio sequence that we had already generated and now put it together with a different technology. Here we used what we call in vivo Hi-C, which is a high quality scaffolding technology that allows us to put together small pieces into very long contiguous chromosomal pieces of DNA. And so from this, we were able to assemble sequences that represented the 18 chromosomes and contained now more than 90% of the total sequence. So we increased the amount of our total sequence that was put together into these 18 large pieces. And importantly, the chromosomes appeared to be put together more correctly than in the previous version. So this is a similar plot now showing the 9 chromosomes from the B and the A subgenomes of quinoa, again with lines connecting genes that are in similar positions on the 2 chromosomes. So for the most part, if these are assembled correctly, we expect most lines to be a linear connection between the 2 chromosomes. We also now see a much more uniform chromosome size in the 2 genomes, and based on microscopy work that we've done, we know that this more accurately reflects the true sizes of the chromosomes. So we have a more realistic representation of the chromosome sizes, and the alignment between the chromosomes suggests that for the most part, these are put together much more correctly than the previous version. So we're quite happy with this new version 2 genome sequence, which we have released publicly, and people around the world are starting to use this genome sequence. Just to illustrate what we can do with these genome sequences, here's an example of using the genome sequence to identify genes that regulate the accumulation of bitter saponins in quinoa seeds. So if you've ever eaten unwashed quinoa, you know that it can be very bitter, and that's due to the accumulation of saponins on the seeds. And so these have to be processed and removed before human consumption of quinoa. We wanted to try to find the genes that regulate the accumulation of saponins in quinoa seeds. And so we had 2 different populations of quinoa that were produced by crossing a bitter variety of quinoa with a sweet variety of quinoa that does not have saponins. And so these are these 2 different populations shown here that we produced by crossing a bitter and a sweet variety, and now producing hundreds of offspring or progeny in each of these populations. And some progeny have saponins and some do not. And what we could do is to look through the genome of the progeny and look for regions of the genome that distinguish all of the bitter progeny from all of the sweet progeny, with the idea being that whatever distinguishes these 2 bitter and sweet progeny might indicate the genes that are different between the bitter and sweet progeny. In both populations, we found the same genomic region, the same region of the genome that was different between all the bitter and all the sweet plants. And when we zeroed in on that region, one gene in particular in that region stood out. This is a representation of that gene, which we call CQ-TSARL1. This gene is known in other species to contribute to the accumulation and to the production of saponins. Interestingly, what we found is that in the bitter parent, so in the bitter varieties that produce saponins, the gene is normal. So the exons of the gene are normally spliced together to produce a functional protein that acts to turn on the pathway to produce saponins and therefore produce bitter seeds. In the sweet parent, however, we found a mutation in part of the gene that appears to affect the way that the gene is expressed and put together in such a way that it produces a mutant protein that is shorter. It's missing part of the protein. And we think that causes this protein to be non-functional, so it cannot turn on the pathway to produce saponins in the seeds. So these seeds lack saponins and are sweet. So we were able to zoom in or focus in on this gene based on the genome sequences that we had. So with that example, I now want to transition briefly and tell you a little bit about some of the genetic resources that we're developing. Because we want to increase production of quinoa throughout the world, we need to introduce new variation for traits. For example, we need to introduce the ability to tolerate certain diseases or certain pests. or the ability to tolerate heat or drought, depending on where we're growing quinoa. One way that we've approached this is to introduce new variation through mutagenesis. And so we can treat quinoa seeds with a chemical mutagen called EMS that mutates the DNA and can produce new traits that aren't in the normal quinoa. And so we treated about 12,000 seeds. We call this the M1 generation. We treated about 12,000 seeds with EMS. From that, we had about 5,000 seeds that survived and produced plants. And so we've been harvesting seed from these plants and advancing them through generations and observing any mutant traits that are present in these plants. And so this work is still in progress. We're still working on analyzing these plants and looking at their genomes. But just to give you an idea of some of the things that we've seen, again, here's what our wild type looks like. This is what non-mutagenized quinoa looks like. And again, this is that Chilean variety QQ74. Here again is a picture of the whole plant and a picture of the leaf structure. Some of the mutants that we've grown are affected in branching. And so this shows mutants that are less branched or highly branched compared to wild type. We've also seen mutants that have phenotypic effects in the meristem, so the part of the plant that produces the flower structures and the leaf structures. So here is a floral head that has an abnormal shape. Here's a plant that is really not producing at all the normal quinoa plant structure. And we've seen many other mutants as well. Here's a mutant that is exhibiting chlorosis or yellowing, which could be indicative of hypersensitivity to nutrient deficiency. We see some mutants where there is a patterning of yellow and green, or in general, kind of a yellowing of most of the leaves except for the leaf veins. And so our objective now is to look through these mutants and find some that are agronomically important, and then try to identify the genes that have been mutated so that we can try to breed some of these new traits into commercial quinoa varieties. Just to give you an example of one mutant that we've done additional research on, just to demonstrate that we can go from a mutant trait or a mutant phenotype and trace that back to an individual gene that has been mutated. Here's what a wild type seedling looks like. When the seedling first germinates, this part of the stem, which we call the hypocotyl, is normally red. We observed a mutant in which the hypocotyl is green. And by sequencing the genome of this mutant, we were able to scan through and identify all the mutations and look at which genes those mutations are in. And again, because we now have a sequenced genome where we know where the genes are and what we think they do, we can quickly scan through all the mutations in this particular plant and find the mutation in a gene that we think is causing this phenotype. And in this case, we narrowed in on a gene called CYP76AD1. This is a gene that's known to be involved in the production of color in plants. In the normal wild type plant, there are 2 exons in the gene that get spliced together to produce a normal protein. In our mutant, in the first exon, there has been a single nucleotide, a single base pair that has changed from a G to an A, and that now introduces a stop codon. And what that means is that the protein stops at this point. It's truncated and does not produce the full protein. So this protein would be predicted to be non-functional and cannot function in the pathway to produce color. This is the pathway that is used to produce color. It starts with tyrosine, and through several different chemical reactions and modifications, it ends up producing a pigment called beta-cyanin, which produces this reddish purple color. The gene that we've identified, CYP76AD1, is known to function in a couple of different steps in this pathway. And so we believe that this mutant gene produces a non-functional protein. And so this pathway to produce the color pigment stops prematurely, and the plant does not produce this color. So this particular trait may not be necessarily agronomically important, but through this work, we've been able to demonstrate that we can identify mutations and mutant phenotypes and trace those all the way back to genes that we believe cause those phenotypes. And this would allow us then to accelerate breeding efforts to try to cross in these mutant genes to commercial quinoa. Current work we're doing now is focused on looking, for example, at dwarf plants. So many quinoa varieties grow very tall and can fall over in the field, especially if there's any wind or hail. We call this lodging. And if the plants lodge, they can't be harvested. And so we are looking for mutants that are shorter, that still produce normal yield and normal seed head, but won't fall over because they're not as tall. And if we can identify these mutants and the genes that are involved in that mutant trait, we can accelerate the breeding of these of new varieties that are shorter and won't fall over in the field. And so I hope this demonstrates a couple of the exciting applications that we have for developing these genetic and genomic resources to try to improve quinoa production throughout the world. So thank you for listening. I hope it was of interest to you. I'm happy to answer any questions that anybody might have. I have my email address shown here and would welcome any questions or comments. Thank you.